Okay, AMP1 students, in this video we are going to start talking about chapter 4, and the title of that chapter is The Biology of the Cell. So the first thing that you need to know is that the cell is actually the smallest living unit in the human body. So if we go back to chapter 1, and remember this was our diagram of the levels of organization in the human body. So we, in chapter 2 and 3, we talked mainly about the chemical level and molecular level. So we get these structures all together, and what they'll do is form a cell. So then like the next most complex structure in the human body. So, but it's this stage where the cell is actually a living unit in the human body. So these guys over here are not living on their own. They don't have any of those characteristics of life that we learned about. So, but the cells do. So they're our smallest living unit in the human body. All right. And so how we study them is under the microscope. So our cells are small enough that we need the aid of a microscope to see them. And so we call this, this is showing a cytologist here looking under the microscope at cells. But cytology is the term for the study of the cells in the body. And I am going to show you a video. And I'll make it bigger too. So here's a little video. And what we're going to look at is see how we can actually take samples of cells from, this is going to be from a dog because that's the video that I liked the best. And we're going to see how we can take samples from a dog or a human, and we can, in just using simple equipment, inexpensive equipment, we can take samples and we can put them under the microscope and we can look at them and even maybe diagnose just from looking at the cells. So in this case, this dog has a swollen lymph node in the neck region. And so you can this video will just show the first few minutes. You can see how the sample is taken and what equipment's used. And it's very inexpensive to do, and it's not very invasive. Most animals don't, and probably people don't mind too much with this procedure. So, okay, so let's watch the video real quick.
He's a suspect lymphoma buddy, and we're going to re-aspirate his lymph nodes. Usually I will not aspirate the mandibular lymph nodes if other ones are enlarged. I'll do the pre-scapular or the popliteal, but these are the only ones enlarged with him, so we are going to do that one. He's drooling a little. Yeah, say hi to everybody. Like the fenestration method with just the needle, no syringe. All right. And then follow me over here. Make some smears. A little bit of a floody one. That's a bad smear. We won't save that one. All right. Now we're going to do, I made a mess here, didn't I, today? Don't tell my girls. We are going to do an aspirate of his lymph node for flow cytometry to help confirm that he has lymphoma and determine if he has B or T cell. So this one I do the needle with the syringe on. Oh, such a good boy. Oh, I know. Good boy.
Okay, so that video was just kind of demonstrating how you can study the cells in a pretty inexpensive and non-invasive way, and to even diagnose some swellings or tumors or things that may be suspicious on an animal or a person. And so they didn't show this in the video, it didn't go that far, but what they would do next is take those slides and stain them. So cells, if we were to look at those slides without staining them, the cells are more, they're not colorful, they're sort of more transparent. And so we really wouldn't be able to see much under the microscope. So they take them and stain them with special stains that the cells will take up, and then those cells will have a certain color, and then we can see those a lot better. So I'm going to show you, in this case, since they thought that dog had lymphoma, this is possibly something that they would see under the microscope. And this is pretty diagnostic of a dog with lymphoma. So there's all these, you can see a lot of cells. There's some red blood cells over here, but it's these big, large cells that are all kind of looking the same, which are a type of white blood cell called the lymphocyte, and they are cancerous. So there's quite a few of them, and they all kind of look exactly the same. So that's probably most likely what the diagnosis was with Ranger, the dog. So now some things can't be diagnosed just simply with cytology. And so sometimes certain tumors, a biopsy is going to be necessary. And that's where they take like the whole tumor or a big part of it, and they will send that whole tumor or part of that tumor to a lab, and they will section it into really fine, thin pieces, and also stain it and look under the microscope. But that way you see not just the cells like you see here, but you would see the entire tissue. So it would look more like the tissues that you guys examined or looked at in lab this week. So and that's called histology whenever they're examining the whole tissue. So some tumors you have to do biopsies to really figure out what's going on. Okay, so that's a little practical information that will hopefully kind of spark your interest in cytology. So I'm going to head back to our objectives. So we talked about the smallest living unit of life, which is the cell. And we defined cytology, that's the study of the cell. And so now we'll finish in this video talk about the organelles and the cell inclusions. And then in the next video, we'll get into the plasma membrane, start talking more specifically about that. So for the organelles and cell inclusions, oh, it's already there. So remember in lab, you guys identified organelles on the cell model, and you also had to know like the general or basic function of each of the organelles. So those are, and we'll talk more about them too in this chapter, but there's other structures that can be located in the cell that aren't organelles. And so one of those is called cell inclusions. And so it's not as complex as an organelle. And so it's more just like a cluster of a particular molecule. So they don't give it an organelle name. They just call it a cell inclusion. So to give you an example, in our skin, we have a pigment called melanin. And that pigment is a molecule, and it's produced by a cell, and then it'll form just in little clusters inside our skin cells. And so they call those, cell inclusions that are inside of our skin cells. So, and then another example would be we talked about in chapter 2, carbohydrates, and the two examples that are most important in the human body is glucose and glycogen. And remember, glycogen is the storage form of glucose in cells, mainly liver cells and muscle cells. And so whenever the glycogen, it's just a molecule that's got a bunch of glucose bonded together. So they call that glycogen that's stored in liver cells and muscle cells, they call it just a cell inclusion. So it's not its own separate organelle. All right. Oh, and then they also talk about triglycerides and adipose tissue. So that would be another example too. All right. So I will see you in the next video, and we'll go into more detail about the plasma membrane or cell membrane of our cells. So in this chapter, this is a chapter where we'll like in lab, we learned some basic general functions of each of the organelles. So in this chapter, we'll go into a lot more detail about exactly how the cell membrane or plasma membrane works. And then we'll also go into a lot more detail about how the nucleus works. So that's what this chapter is primarily going to focus on. So, okay, I'll see you in the next video.